Welcome to this lecture on prokaryotic protein synthesis. Today, we are going to explore how bacterial cells read their genetic blueprints and build the molecular machines that keep them alive. This process is not just a biological marvel, it is the very foundation of how life sustains itself and adapts. By the end of this session, you will understand the mechanics, the energy costs, and the incredible speed of this cellular factory. Let us dive in. To begin our journey, imagine building a complex Lego castle using instructions written in a foreign language. You need a factory to assemble the pieces and a translator to hand you the correct blocks. In a bacterial cell, the messenger RNA provides the instructions, the ribosome is the factory, and transfer RNAs are the translators. Before the factory can even start, these transfer RNAs must be armed with the correct amino acids. This crucial step is called charging, and it is performed by an enzyme called aminoacyl tRNA synthetase. This process matters immensely because proteins do almost everything in a cell. If the charging step lacks precision, the resulting protein will be defective, which is why this step is highly regulated and targeted by many antibiotics. Now that we understand what transfer RNA charging is, let us look at the energetic cost of this process. The formula for the energy required to charge transfer RNAs is E charge equals 2 times N. Conceptually, this comes from the fact that attaching a single amino acid to its transfer RNA breaks down one molecule of ATP into AMP and pyrophosphate. Biochemically, this is equivalent to consuming 2 high-energy phosphate bonds. In this equation, E charge represents the total number of high-energy phosphate bonds consumed, and N is the total number of amino acids in the final protein. As the length of the protein, N, increases, the energy required increases proportionally. This shows just how expensive it is for the cell to prepare the building blocks for protein synthesis. Before we move on, let us see this with a concrete example. Suppose a bacterial cell needs to synthesize a small structural protein that is exactly 50 amino acids long. We are asked to find how many high-energy phosphate bonds are consumed strictly during the initial transfer RNA charging phase. We chose our charging energy formula because it directly relates protein length to the initial energy investment. We start by identifying our given value, N equals 50. Next, we substitute 50 into our equation, multiplying it by 2. This gives us a final answer of 100. Physically, this means that exactly 100 high-energy phosphate bonds are cleaved just to prepare the materials for this small protein. This massive toll on the cell's ATP reserves explains why bacteria tightly regulate when they build proteins. Having prepared our building blocks, we can now explore how the factory actually assembles them. Translation is divided into 3 distinct stages: initiation, elongation, and termination. Think of it like building a train. Initiation is placing the engine exactly at the starting line, which in bacteria is the Shine-Dalgarno sequence on the messenger RNA. This ensures the reading frame is perfectly aligned. Next is elongation, where we bring in train cars one by one. Charged transfer RNAs enter, a peptide bond is formed, and the ribosome translocates, or moves forward by one codon. Finally, termination occurs when a stop codon is reached. A signal tells the ribosome to stop, release factors enter, and the fully built protein is released to go do its job. This highly structured mechanism is universally conserved across all bacteria. To visualize this assembly process, let us examine the prokaryotic ribosome during the elongation cycle. In this diagram, You are looking at the complete ribosome clamped around the messenger RNA strand at the bottom, which is read from the 5' to the 3' direction. Notice the three distinct chambers inside the ribosome: the A site, the P site, and the E site. The A site is where a new charged transfer RNA enters, carrying the next amino acid. The P site holds the transfer RNA attached to the growing polypeptide chain, shown here as a string of beads. After the bond forms, the ribosome shifts, and the empty transfer RNA exits through the E site. This diagram beautifully illustrates the continuous, cyclical nature of protein synthesis. Now that we have seen the full cycle, we must consider the total energetic price tag. The formula for the grand total of energy used to synthesize a complete protein is E total equals 4 times N. Conceptually, this accounts for the entire journey of every single amino acid, from its initial charging to its final placement in the protein chain. In this formula, E total represents the grand total of high-energy phosphate bonds consumed, and N is the number of amino acids. If the length of the protein increases, the total energy required scales up by a factor of 4. This means that building a protein is an incredibly heavy financial burden for a bacterial cell, requiring a massive continuous supply of ATP and GTP to keep the ribosome moving. Let us apply this total energy concept to a real-world scenario. Imagine a bacterium detects a new sugar and must synthesize a vital metabolic enzyme that is exactly 300 amino acids long. We need to calculate the total number of high-energy phosphate bonds spent to completely translate this enzyme. We choose our total energy formula because it accounts for charging, binding, and translocation all at once. We start with our given length, N equals 300. We substitute this into our equation and multiply by 4. This gives us a final answer of 1,200 high-energy phosphate bonds. Physically, wasting 1,200 energy equivalents on a protein the cell does not need would rapidly deplete its reserves. This perfectly illustrates why gene regulation is critical for survival. Now that we have used this formula, let us understand where it comes from by breaking down the costs. This chunk of our derivation establishes the energy spent on every single amino acid added to the chain. First, as we learned earlier, charging the transfer RNA consumes 2 high-energy phosphate bonds. Next, during the elongation phase, bringing that charged transfer RNA into the ribosome's A site requires the hydrolysis of 1 GTP molecule, which equals 1 bond. Finally, moving the ribosome forward by 1 codon, known as translocation, requires another GTP molecule, equaling 1 more bond. Adding these together, the mathematical reasoning shows that each amino acid strictly costs 4 high-energy bonds during the main cycle. This builds our intuition for the core expense of translation. So we have proven that the core cycle costs 4 bonds per amino acid. This brings us to the final accounting of the entire process. If we multiply the per amino acid cost by the total number of amino acids, N, we get 4N. But what about the start and end of the process? The initiation step uses 1 GTP molecule to assemble the ribosome. The termination step also uses 1 GTP molecule to disassemble it. In our simplified mathematical model, these extra costs at the boundaries balance out perfectly with the mechanics of the first and last amino acids. Therefore, the mathematical reasoning simplifies beautifully, leaving us with the final proven result. The grand total energy cost is exactly 4 times N. This elegant simplification makes it easy to calculate the cellular burden of any protein. Having established the mechanics and the cost, we must now look at the speed of this machinery. Imagine a highly skilled typist copying a book at an astonishingly fast and steady pace. In bacteria, like E. coli, the ribosome translates at a rate of approximately 15 to 20 amino acids per second. Because prokaryotes lack a nucleus, the ribosome can attach to the messenger RNA while it is still being synthesized by the RNA polymerase. This phenomenon is called coupled transcription translation. This speed matters immensely because it is a matter of life and death. If a bacterium is exposed to a toxic chemical, it must synthesize protective enzymes immediately. If translation were slow, the bacteria would not be able to adapt, reproduce, or compete in their environment. This incredible speed leads us to a simple way to calculate how long protein synthesis takes. The formula for translation time is T equals N divided by R. Conceptually, this is just the classic physics equation of time equals distance divided by speed. Here, the distance is the length of the protein, and the speed is the ribosome's rate. In this formula, T represents the total time in seconds, N is the total number of amino acids, and R is the rate of translation. If the length of the protein, N, increases, the time required to build it naturally increases. Conversely, if the ribosome works faster and the rate R increases, the total time required will decrease, allowing the cell to respond to its environment much faster. Before we conclude, let us see this kinetic formula in action. The enzyme beta-galactosidase is used by E. coli to break down lactose. It is a massive protein that is approximately 1,000 amino acids long. Given a rapid translation rate of 20 amino acids per second, how long will it take to synthesize one complete molecule? We are given our total distance, N equals 1,000, and our rate, R equals 20. We chose the translation time formula because it directly relates these variables. By substituting 1,000 for N and 20 for R, we divide to get a final answer of 50. Physically, this means that in less than 1 single minute, a bacterial cell can completely synthesize a massive, complex enzyme from scratch, allowing it to digest new food sources almost immediately. We have covered a tremendous amount of ground today, so let us summarize the most important ideas. First, prokaryotic protein synthesis relies on the extreme precision of transfer RNA charging to ensure the genetic code is translated accurately. Second, we saw how the ribosome orchestrates the 3 stages of initiation, elongation, and termination, acting as a highly efficient molecular factory. Third, we learned that this process is incredibly energy-intensive, costing the cell 4 high-energy phosphate bonds for every amino acid added. Finally, the unique ability of bacteria to couple transcription and translation allows them to build proteins in mere seconds, giving them a massive evolutionary advantage. You now have a profound understanding of the microscopic machinery that powers bacterial life. Great job today!